Hello, I'm Jordyn Sisovsky, and our research covers the advanced alternatives to antibiotics, investigating bacteriocin activity against MRSA and Group B strep. Bacteria produce antimicrobial peptides called bacteriocin that inhibit the growth and/or kill other bacteria. Bacteriocins are produced naturally and are the microbe's defense mechanism to outcompete other bacteria. Harnessing these bacteriocin activity will reduce our dependence on antibiotics, thus decreasing antibiotic resistance and associated human mortality. This study serves as an investigation into bacteriocin against methicillin-resistant Staphylococcus aureus, or MRSA, and Group B streptococcus, or GBS. The experiment also includes improved bioprospecting techniques and antimicrobial activity assays. We chose 5 species of bacilli isolated from environmental sources that were used as our antimicrobial peptides producers. We specifically chose bacilli because of their already well-documented bacteriocin activities. We also isolated lactobacilli from an infant probiotic called Flora to test its effectiveness against the pathogens. To isolate the lactobacilli, we serial diluted a sample of the probiotic and then subcultured it onto 2 different kinds of media, BHI and MRS. To get a baseline for our pathogens, an Alto-Cirillo microplate reader was used to compare the growths in aerobe and anaerobic conditions. Bacilli whole genome sequences were screened through the anti-SMASH database, indicating whether they had homologous genes to known bacteriocin genes. Next steps in the experiment, we will grow up the bacilli and lactobacilli in a 96 well plate, and then using a microplate stamp, we are able to transfer the microbes from the 96 well plate onto an agar plate. We then overlay the now stamped agar plate with an agar that has been inoculated with one of the pathogens. This is then incubated for about 24 hours, and during the incubation period, we begin to see areas of clearing, indicating that the bacilli that was stamped in that area has inhibited the growth of the overlaid pathogen. While this study is still underway, we anticipate zones of inhibition from all bacilli species against our pathogens MRSA and GBS. We also expect a protective effect from the lactobacilli species isolated from the probiotic. Current experiments also include bioprospecting novel organisms and assays for their bacteriocin-producing activities. To do this, we serial dilute the specimen and plant them onto different agars. We then pick, purify, and characterize these colonies and have their genome sequenced to determine if they are novel. So at its core, this research emphasizes bioprospecting, exploring nature's ability to produce bacteriocins to combat the growing epidemic of antibiotic resistance. The hope is that these bacteriocins could pave the way for targeted therapies, reducing the collateral damage caused by broad-spectrum antibiotics.